My research lab here at the MD Anderson Cancer Center has specific areas where you do specific things. So there's a room in which we grow cells, and we have to be able to grow cells to understand how cells work. And we grow different sorts of cells, leukemia cells, brain tumor cells, and these are models for how the disease actually works. So these are the things that we work with on a daily basis. And these cells are growing in incubators. So we have several incubators that have the cells that are in dishes. These dishes kind of look like flasks, maybe not the type of flask that you've seen in school so far, because they're flat. And they have to have a flat surface area for there to be a solution in which the cells are growing. Also, the solution that these cells are growing in is full of nutrients that things like bacteria or yeast might want to grow in, and we don't want those things to grow with our cells. So we have to have an area where we can work sterilely with these cells. So this room that's called our cell culture room has incubators, which the cells live in, and then there's an area that has a sterile environment where air is being blown through to keep it sterile, and we work inside that area. So all of the manipulations with the cells are done in this sterile environment. So that's the cell culture room. If you walk out of the cell culture room, you enter the larger part of the lab, and that's where all of the protein work occurs and all of the DNA work occurs. And so those are different pieces of the cell which we study extensively. And in order to do different manipulations with those cellular components, we have to have different solutions. We have to have specific machines that can separate out pieces of DNA or separate out pieces of protein. So all of that equipment is in the rest of the lab. But you also have to have a place in which you can do your experiment with these pieces of equipment. So every person in the lab has a lab bench. And the lab bench just looks like a counter with a stool, and you'll frequently see a student or a postdoc working at that bench, usually with a handheld device that can measure very small quantities of liquids, and that device is called a pipette. And the pipette is able to transfer and measure small amounts of liquid, and the liquid will contain solutions that have protein or DNA or different cellular pieces. So a lot of the work that we do is invisible to the naked eye, and you have to take very specific types of pictures of it. You have to look at things under a microscope to actually see the results that we generate.